Many RNA molecules fulfill their function by interacting with other molecules. Two RNA molecules can pair if the respective sequence parts are complementary. Complementary in this case means that each C can pair with each G and each A can pair with each U. IntaRNA can detect potential interactions between two RNA molecules and can estimate how strong these interactions are. For this, IntaRNA also takes into account that sometimes the existing structures need to be changed in order to allow the interaction.
One great thing about IntaRNA is that it is very fast. So this means you cannot just take two RNAs and check whether and how good they interact, but you can actually scan entire genomes for potential interaction. And how this works with the help of our web server, I'm going to show you now.
Okay, so here you see now the Freiburg RNA Tools web server that you can find at rna.informatik.uni-freiburg.de. And I already opened here the website of IntaRNA. And IntaRNA you can use here basically in two different ways. The simple and fast way is the one where you enter here in this field a non-coding RNA in FASTA format, and here a set of potential mRNA candidates that it can be or that it can interact with, and then IntaRNA analyzes all the potential interactions of the ncRNA to the mRNAs here. There's also a much more powerful variant that I want to show you now, and that is that you do not give a set of mRNAs manually, but that an entire genome is scanned. So you click on this here, and then here you do not give an mRNA, but instead you give a genome accession number and which areas of the genome should be searched. And the example I prepared is that I want to search for potential interactions of the MIC-F RNA that is described on this Wikipedia page here. So of this MIC-F I also already prepared the sequence, so I paste the sequence here, and I want to search in the E. coli genome because here it is written that it is found in Escherichia coli. So I need the accession number of the E. coli genome, and this you can find here at the NCBI pages of genomes. So here you can see I've opened, for example, the site of all possible bacteria, and here you have their names and here their genome accession numbers. And if you scroll down the list, you will find that the E. coli has the following accession number here. And now we need to specify which areas we want to search, and usually non-coding RNAs interact at regions around the start codon. So you can also search for regions near the stop codon, but we go to the start codon and choose in this case the area from 100 nucleotides upstream to 50 downstream of the start codon. And here you can set the seed. This is the length of the region that should exactly match at least of the two RNAs. 7 is a good and the default parameter here, so we leave it as it is and click on Go. Now the genome is retrieved from NCBI and then it is searched by our server. This takes a while. In this case it takes around 30 minutes. Okay, here we have now the final result. You see that the job was processed or submitted at around 7 o'clock, and a little bit more than 30 minutes later it was finished. So this is the time that IntaRNA needs to search or to scan, for example, the entire E. coli genome. Here's then the result, and you can see here in total we have around 4,000 potential interactions. So this is quite a lot, and this does not mean that all of them are really true interactions, but they are sorted according to their quality. So the first ones in the list are the most likely ones to be interacting. And yeah, if you look at this list, I want to highlight here specifically the third one, OMPF, and that is because if we go back to the Wikipedia page, you can see that this mcRNA actually interacts with the target OMPF. So the correct interaction partner was found in this list already at position 3. So quite a good quality. And if you click on such an example, you see here the interaction itself, which part of the two is interacting. And let me click on it again. You can automatically go here to this page of NCBI where the respective gene is described. So by that you can really conveniently browse all the potential interactions and see which of them are interesting for you. Okay, this was the overview over IntaRNA. I hope this was helpful for you.